Okay, cell cycle control and mutation. Now remember, genes, they don't really get turned on or turned off. It's more like a rheostat or a dimmer. But of course, you could get a gene a mutation that causes a gene to stop working entirely. So its protein product is no longer made. Or the same sort of thing could happen. A gene is suddenly turned on and it's, let's say, in the promoter region, and it's causing a gene downstream to be expressed all the time when it shouldn't be. So you can have a mutation that, in fact, no longer codes a protein that's necessary, or alters the function of a protein, or have little or no effect. Okay. Let's see. Okay. I forget. I think it'll answer a question in the next one. Oh, proto-oncogenes. Yeah, I don't like to call them that. Okay, that kind of gives you the impression that our body is literally riddled with genes that are likely to go crazy, and they're not. Okay. Basically, these genes are normal parts of the cellular machinery for replication, and with a certain mutation, they can no longer do their job properly. Now, in fact, instead of calling them proto-oncogenes, we'll call them tumor suppressor genes. And the most common mutation is one that inactivates a tumor suppressor gene. In other words, that tumor suppressor system is no longer working and stopping cells from going nuts and dividing uncontrollably. In fact, I'll just write that down. Okay, so here are a bunch of different cell cycle checkpoints that I talked about earlier. These are stop points. So let's say police set up a dragnet and they stop people coming through because they're looking for someone who did something dastardly, like a pirate does. These checkpoints, as long as the cells pass muster in terms of their cell cycle, everything's good. But if they don't, if there's this individual who's done crimes, he'll get stopped. So at that checkpoint, he'll get found out, captured, and that'll be the end of it. There are also checkpoints at the end of G2, and then at points during mitosis. So tumor suppressor genes make proteins that stop cell division. So these checkpoints actually stop cell division, but it's only for certain cells that show craziness and other cells that don't. Okay, this is something that we saw earlier, a dividing mitotic cell. So this is mitosis. Of course, this is the nucleus where it all happens for eukaryotic cells. So right here, this is a non-mitotic cell in G1. Okay, actually. And this is actually this cell right here is the one that's undergoing mitosis. You can actually, you can actually see the chromosome starting to condense. Okay. So the G1 checkpoint. Now normally a cell will stay in G1 most of its life. Only sometimes will it get a signal to replicate. So the G1 checkpoint checks actually if cell division is actually necessary. So if the cell division isn't necessary, but you have a cell that appears to want to go into the S phase, the suppressor gene will stop that from happening. Are there certain growth factors present that shouldn't be on, but are, that control cell replication? Is the cell large enough? So a cell has to have enough resources to actually, you're making extra copies of the DNA. That's expensive. And then for the G2 phase, that's when you're actually making all the other stuff that you need for the cell to actually have enough resources to divide into two. And then are sufficient nutrients available. Now, was the DNA in checkpoint 2, it checks, was the DNA replicated properly? It turns out that a lot of eukaryotic cells have this system where after DNA is replicated, and you've got a double-stranded DNA again, a protein will go along the chain to make sure that it was actually copied properly. So, and then is the cell large enough? In other words, during G2, the cell should have enough nutrients to actually be able to undergo cell division so that the two daughter cells are actually healthy. And then the metaphase checkpoint are all the chromosomes attached to the microtubules, which we'll look at in a cell that's actually dividing. Okay, one thing I do want to do is, Okay, I just want to go over the cell cycle again because it was a lot, and the more we go over it, the better. So, G1, S, and G2 are what's called interphase. Okay? And then mitosis is the last part of the cell cycle. Most cells remain in G1 for their whole life, for most of their lives. Okay? It's only when they get a signal to actually divide that they then go into interphase. They go into S. So we start in G1, and interphase is G1, S, and G2. So we go into S and we duplicate every gene or every chromosome. And what I mean by that is we have these homologous chromosomes, one from mom, one from dad. Those homologous chromosomes are, or the chromatids are duplicated so that then we have a chromosome, maternal chromosome that has two copies of itself, two chromatids. We'll go back over that a little bit. In fact, I probably should do it right now. So bear with me. Okay, so This is S. This is when the chromosomes get duplicated. And, okay, so again, S, this is where it all duplicates happens. So we start out with one copy of the maternal, one copy of the paternal. That's a homologous chromosome. During S phase, we duplicate that. We have an extra copy of the maternal and an extra copy of the paternal, and they're tied together right in the middle. Okay, and this is what it looks like. And the centromere is right in the middle there. Here, I can do this. Here's the centromere. Okay. Now, it's condensed. And the maternal duplicates so that it has 2, and then the paternal duplicates and it has 2. So now there's twice as much DNA as normal. Okay, so let me go back. We go into mitosis. So here's a little bit better view. Okay. This is our interphase, right? Let's say we've duplicated the DNA in S phase. Then we're into G2. So we enter prophase. Oh, and here, let me show you actually. Okay, so here's G1, right? This is the S phase. So we've got single chromosomes here, and now we have double. And then we've got G2. Let's see, right in here. And then finally we go into mitosis. Okay. So all this is interphase. Okay. And now we finally go into mitosis, and we'll go back to this one so it shows it a little bit better. Okay. So here's our interphase, right? Now we're in prophase. These are sister chromatids and duplicated chromosomes. So let's go. Here's the maternal chromosome and the paternal chromosomes. Now, when the DNA isn't duplicated, all we have normally is okay, and then we have okay, and maternal. Now, what's happened here is that we've duplicated this and this, and now we have okay. These are maternal, these are paternal. Okay, now okay, so we have prophase, This is where the condensing starts. You also start to see these centrioles start to form right here and right here. This is where these fibers are going to pull apart the different, each individual chromosome. So one of these will go here, one of these go there, so on. So when they finally get to along the metaphase plate, and this is the metaphase plate, okay, there's these spindle fibers that then come down, grab part of the centromere, so each one has a centromere, and then during anaphase, these are pulled apart, and they go to one side or the other. And finally, we have telophase, where the cell membrane has reformed, the nucleus has reformed, and then, of course, we finally have cytokinesis, which is, this is telophase, and there's cytokinesis right here, where we have two different daughter cells. And they now have one maternal and one paternal. Any questions? Okay. Now you can go over this yourself, but I've pretty much broken it down into what mitosis actually accomplishes. There's also a lot of stuff. If you go to the chapter for this cell cycle stuff, it'll take you through it all as well. Okay. Let's see, what did I want to say? Oh, okay. Okay. I didn't have that recorded, so I'll just record it again. Okay. This is the gene. This is okay, region. And then promoter. Okay, good. Okay. Okay, now we're back to cancer. So, self-suicide is called apoptosis. Here we've got a cell. It's looking a little hinky. It's saying, I don't want to die. I'm going to keep making copies of myself so I go on living forever. There's actually some cells from a tumor cell from a woman that was harvested back in the 50s that is still a cell line that's been copying itself over and over again in the lab. So they got these tumor cells, they grew them in a petri dish, and they've actually created cell lines from this tumor cell to actually investigate cancer. So there's a normal cell, as you can see, with all these different, let's see, all these different proteins, right, on the cell. And that's how immune cell can actually detect whether a cell is cancerous or not if it has proteins that are of a different type. And then here's an apoptotic cell, one that's killed itself. And here we see it falling apart. God, it is late. Let's take a break. Is that okay? Yeah, I think so. Okay. I'll go back over this when I actually put it online. So 3 checkpoints. BRCA is this tumor suppressor gene. If it doesn't do its job, the cell can divide, and as it does that, it might accumulate mutations and then eventually lead to cancer. Okay. So unregulated tumors are a problem. Now, some are benign. That means that cells are dividing, and they form a tumor, but they don't invade adjacent tissue. Okay? No adjacent tissue. And the thing that actually stops a benign tumor or any cell from invading adjacent tissue is something called contact inhibition. So the tumor cells come in contact with an adjacent, let's say, tissue type, and other cells, and it stops growing right there. Okay? It doesn't mean that benign tumor can still cause problems, but it's not an invasive cancer yet. Okay. Now, when cancer cells lose contact inhibition, it means they can invade adjacent cells. So let's say you've got liver, and you've got some cells that start dividing uncontrollably, and it forms a tumor of a bunch of different of these cells. It's not invading the adjacent cells, so it's a benign liver tumor. It may still have to come out. They may still have to do something about it. But the thing is, since wherever it comes into contact with adjacent liver cells, it stops. It's pretty restricted in an area, and it's not going to grow crazy, really large. Now, if the cell loses contact inhibition, these benign cells can actually then invade, proliferate into the adjacent liver cells. It's usually the liver duct cells is where liver cancer starts. Okay. Salute.
Can alcohol, alcohol gives you liver cancer, right?
What?
Drinking.
Oh, okay. So alcohol or anything like that, it causes cirrhosis of the liver. Okay, so it can cause cancer, but it's kind of a byproduct of damaging all these cells in the liver. So you've got all these damaged cells which have mutations. Some of them are likely to, they just might stay, damaged. The liver doesn't work as well. Those cells don't do their job, but it doesn't necessarily mean cancer. But some can. Similarly, if you have, what is it? Let's see, hepatitis C. That's a viral hepatitis of the liver cells. Hepatitis C often causes cancer, I don't know with a percentage of the time, but it certainly increases the prospect of developing cancer in those cells. So most people, and it also causes cirrhosis of the liver, but not cirrhosis like from alcohol. It's from the actual viral stuff, okay? And then, so now they've got a new treatment. It's pretty expensive, but where they can actually get rid of and cure hepatitis C, okay? So that's a big deal. Yeah?
Can you leave benign tumors alone?
So a lot of times you can, because since they're usually surrounded by other cells, and they run into them, they stop growing. So it kind of restricts how big they are. But sometimes they can cause problems. You can have a benign tumor on, let's say, the pituitary gland, which affects hormone production, and it'll cause all sorts of problems. So that might actually have to be removed. Okay? Okay, so here's a little model of what happens. We've got a tumor. It's either benign. Usually benign tumors, nothing has to be done, but sometimes they have to be taken out. Or you actually have malignant cancer, which is invasive, because the cells have lost contact inhibition. In other words, when it runs into adjacent cells, it actually invades them, okay? And starts growing within that adjacent tissue, making more and more tumor.
Another question. Can benign tumors turn into malignant?
What?
Can benign tumors turn into?
Malignant?
Yeah,
I imagine they could if they generate enough mutations.
Oh, okay.
But I don't think they normally don't, but
So it's one or
but
the
it could
other?
be, yeah, but I'm not really sure about that. It sounds like, I'm just thinking about it. Certainly as those benign tumor cells replicate, they might generate more and more mutations, at which point some of those might actually lead to the loss of contact inhibition, and then those cells will then become cancerous.
Oh,
Okay? Now, there's also the worst kind of cancer, usually, is the kind of cancer where it can travel from one part of the body to the other. Now, normally, when we have a tumor, let's say in the ovary, okay, it's usually all these different other cells in turn, like these ovarian cells, right? They're all anchored in place. So they stay where they are. But you can lose anchorage dependence. And then, when that happens, these cells can now wander. They can get into the adjacent lymph nodes or bloodstream, and they can get carried all over the body, where they can stop maybe at certain places and start forming secondary tumors from the primary tumor. So let's say the primary tumor was an ovarian tumor. Now you're going to start seeing ovarian tumors popping up all over the place. And those ovarian tumors, they'll probably be doing what ovarian cells do, which is make estrogen and other hormones. And so you're going to see some metabolic abnormalities when they do a blood screen. Yeah.
So how does the leukemia work where you don't necessarily have a tumor, but
Oh, okay. So, it's a blood tumor, but usually it starts in the marrow. Okay, so the marrow cells are stem cells that then differentiate into different kinds of blood cells. If they differentiate into red blood cells, then you usually have things like aplastic anemia, thalassemia, some stuff like that, where you have a bunch of immature red cells being made. If it's actually white blood cells, where there's a problem, then it's differentiating into these white blood cells that are the immune system, and they tend to clog different organs because they build up their immature white cells. Okay, so that's the part. So it's a blood. Leukemia is a blood tumor. Hodgkin's disease is a lymph tumor, but it doesn't cause solid tumors. It just circulates in the lymph, whereas lymphoma actually causes lymph tumors. Okay, so here we go. We've got a loss of anchorage dependence. It allows these cells to not only move around, but also leave and get into the blood or lymph system and be carried around to cause additional secondary tumors. Now, one other thing. All this time that this is going on, these cells are still dividing, right? They are probably still generating new mutations. So the initial cancer, if you charted it along a timeline, you'd see additional mutations start popping up. And then, of course, when you have metastatic tumors elsewhere, they're probably going to have their own genetic signature. Okay? Kind of like human migration. So humans started in Africa, and then they started spreading out and creating populations across Asia and Europe, and then down into Southeast Asia and Australia, and then finally into North America. So individual populations started popping up of all these different people. Well, the same thing is happening with this. So now we have cancer regression. Malignant, non-metastatic, it means there's only a primary tumor. It doesn't mean that it won't kill you, but at least it's not spreading all over willy-nilly. If it is traveling, then you've got metastatic cancer, and the chances of stopping it are reduced. But not necessarily, because if you actually have one of these therapeutic agents that interrupts a specific pathway for the cancer cells because of the mutations they have, then that'll cure the cancer. Okay. So, normal cell division, potentially cancer cells. So here we have a benign tumor. That means there's contact inhibition. And here we have a malignant tumor that's invasive. And then finally we have this malignant cell that actually can break away, lose anchorage dependence, and actually travel to different parts of the body. Okay. There's also, in terms of top hat, there's that extra question about BRCA, so you can go ahead and answer that. I'll leave it open. But moving on. So there's a bunch of different things that can actually cause cancer. There are chemicals that are carcinogens. There are chemicals that are also mutagens. They can cause mutations that then might end up being cancerous mutations. Some of these chemicals that can do that, some are natural, some are not. So there was, I remember in China there was a food where the food grew mold on it. Kind of like what we do with blue cheese, or a lot of cheeses where you end up growing different kinds of mold on it. And unfortunately, this mold had created a toxin that was a carcinogen. So it's natural. And it actually produced lots of esophageal cancers. Okay? People don't usually eat it anymore. It was kind of, when they were all peasants and stuff. And so it's really gone down in the incidence that it used to show before. But it's a natural carcinogen. Okay. Smoking, obviously. Tobacco smoke has all sorts of different chemicals in it, not just nicotine. Now, nicotine, it turns out there's wild tobaccos that are literally hallucinogenic. You can find them in South America. And if you smoke that stuff, it has enough nicotine to actually kill you. Okay? So if you take enough nicotine, it can really damage you. But in cigarettes, there's enough nicotine to give you the rush, but not enough to kill you. But there's a whole bunch of other stuff in tobacco smoke that's burned that, those other chemicals can cause cancer. Okay? So, of course, you can always vape, I guess, if you want to. As they either add nicotine to it or they don't, so people can vape if they want. It's not smoke, it's actually a vapor. So, and who knows how easy it is. If it's dangerous or not. It's certainly much less dangerous than actually smoking cigarettes. Okay. And so, of course, tobacco causes a lot of lung cancers, small cell carcinomas, and then it also, especially if you chew tobacco, it can cause oral and esophageal cancers as well. It's also from smoking or chewing. So risk factors. Let's see. Sex is a risk factor. You want to look cool, so you smoke a cigarette. And you want to look cool because of sex, right? You want to have sex. So you sit around and, You smoke, look cool, eventually you have sex. Frankly, it doesn't taste very good. So I just leave out the smoking and just have sex. Much better, okay? Now, there's also, it turns out that cigarettes can also cause cancer of the colon, because not only you're inhaling, but you're also swallowing these carcinogens that then end up in the colon and are carcinogens, so they cause cells to mutate in the same way. Now, there are all kinds of different carcinogens. UV light is actually a carcinogen. But usually when we talk about carcinogens, we talk about chemicals that cause cancer. But certainly UV light, you get melanoma or X-rays. Ionizing radiation like gamma rays, either from a nuclear meltdown or a nuclear bomb going off. Carcinogens, tobacco smoke, all sorts of different things in nature that can actually cause cells to mutate and to become cancerous. A lot of what we have, like common poisons, like, as I mentioned, let's see, cyanide chemicals. So if you probably know this, if you eat the pits of apricots or peaches, the pits are very bitter. They actually smell a lot like almonds. Well, they have a really high concentration of what's called cyanogenic glycosides. These are chemicals that when they come into contact with a enzyme, break down, and then release hydrogen cyanide gas. Okay, that alone can get you really sick, but it also turns out that depending on levels of cyanide, not only can it kill you, but it can also cause cells to become cancerous as well, mainly because it interrupts the whole cell respiration cycle that we talked about earlier for aerobic respiration. I guess a poor diet can result in cancer. It looks like the more meat you eat, the higher your chances of colon cancer. But, you know, I would treat a ribeye steak more like a fruit. And if you think about it that way, then you won't get cancer from it, okay? This is hanging there from a tree. It's a ribeye tree. If you've ever seen one of those, it's amazing. Yeah, okay. So, low-hanging fruits and veggies. Okay. Now, supposedly, fruits and veggies help protect because they stop antioxidants, which are these free radicals. And so, if you have these, if you don't have enough antioxidants that clean up these free radicals, you get problems. Whether that's actually true or not, I don't know if it's actually been demonstrated. Genetic predisposition. You might inherit a mutation from your parents that makes you more likely to be susceptible to carcinogens and cancer. Lack of exercise. So, that can cause problems. Keeps the immune system healthy. Our increasing age. Now, increasing age is a double whammy. We get tired, we get lazy, and we don't want to exercise. So that's not good. And then, of course, our tumor suppressor genes aren't really, they're kind of like old cops. Old cops can't chase down criminals like young cops can. Okay? So, as you age, you get usually more susceptible to some kind of cancer. Okay. Let's see, where are we? I think we'll call it a day, and then when we come back, we'll watch this video.